You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts, and I have Whitney Stoppel. She's a professor at the University of Florida, head of the Stoppel Lab, named after her, and we're going to be talking about her work there. So, Whitney, thank you for coming.
Thank you.
Yeah, the subject is a bit complicated, so it's probably better if you explain it, but can you talk about the nature of your work and the basics of it?
Sure, yeah. So at the University of Florida, my lab is really interested in understanding and designing natural materials that hopefully eventually a surgeon will be able to utilize to aid in repair of either injured or diseased tissue in a patient. And so we're really interested in being able to design these materials such that they have different properties that kind of match the wound healing rates of our patients. So a lot of times patients who have diabetes or, have had some other secondary complications, recent surgeries, things like that, can have slower wound healing rates than, say, a healthy patient or even a young child. And so we're really interested in understanding how we can build and create materials that will meet these kind of different timescales that our patients will present with in the clinic.
Well, I mean, there's so many tissues in the body and opportunities. So what kind of tissues and what's the focus of it initially?
Yeah, so I agree that the body is quite complex. And so, we're interested in starting with looking at things that are kind of more familiar from my background. So looking at things like skeletal muscle or cardiac tissue, which are kind of tissues that have an elastic nature to them. And as patients age or during disease, this elastic nature of the material or of the tissue itself kind of changes. And so we're interested in being able to build materials and design these natural biomaterials that have this elasticity and structure that matches what healthy tissue will look like.
Well, what kind of tissues are you, how far along are you? Have you picked a particular tissue, and are you on the way towards creating it or engineering it or altering it?
Well, so right now the lab is new. We just opened our doors and took our first PhD students in October. And so we're kind of just getting up and going and transitioning some of the work that I had been focused on during my postdoc studies at Tufts University and translating some of the same concepts and ideas, but for different tissues and strategies at the University of Florida.
So what's the method by which you would construct tissues or alter them? Is this like 3D printing, or is it taking an existing piece of tissue and culturing it a certain way?
Yeah, so there are many different strategies that you can generate three-dimensional structures in the lab. 3D printing is definitely a new and hot topic. However, one of the materials that we're really interested in is silk fibroin, which is a natural biopolymer that can be extracted from the cocoons of a silkworm. And so we're actually able to take these cocoons and wash them and actually boil them and break the polymer or protein structure down into a solution format, kind of like a viscous solution like honey and water, and then take this solution and we can cast our scaffolds that we'll use to eventually grow muscle tissue in different molds. And so then these molds, once the polymer solution is in the mold, there are a few different ways that we can kind of set the polymer structure through freezing and removal of water, and then finally through basically processing the silk protein and turning it from something that's soluble in water into something that is not soluble in water. And so once we've converted it into a format that isn't soluble in water any longer, we can then have this fully formed scaffold that we can utilize in our tissue structures.
So will an entire piece of tissue be grown outside the body and then inserted, or can you do this in vivo?
So actually, that's a really great question. So from a biomaterials and tissue engineering standpoint, a lot of people are really interested in doing both. And so I think that way down the road in the future, it will be great if we can create tissues outside of the lab that have living cells in them that potentially came from the patient who was injured and then cultured outside of the body and implanted to help that patient, with their wound healing. However, right now, I think that there are a lot of kind of clinical hurdles to doing that and being able to make sure that the materials with cells in them are really sterile, that we don't induce any sort of contamination or in that case, like something that might cause an infection in our patient, or even introduce cells in a way that could lead to abnormal growth or, scar tissue formation. And so for us right now, we're really interested in being able to make these materials in an acellular format or a format without any cells in them. And so we'll put cells in them to understand how the cells respond and to do fundamental analyses of kind of how the cells like or dislike almost the material. But the goal is right now to develop an acellular platform that a surgeon could utilize, because these are much easier to sterilize. They're much easier to kind of translate into a surgical environment. They're easier to store. You don't have to make them right away. And so there's a lot more flexibility in kind of that clinical translation aspect.
Well, how do you know what the morphology of a tissue should be, or how would the cells know which direction to grow in, when to stop, what shapes to make, and all that?
Yeah, so that's basically one of the main thrusts of our research. So as we're looking at these materials, we like to think about kind of how the cell is going to perceive the material, how the cell or the tissue is going to digest the material, and over what time period that might happen. And so for skeletal muscle kind of repair or rehabilitation, we're really interested in trying to get at the time scales at which that repair happens in the body, and then be able to tune our material to kind of degrade and disappear over that same time scale. So what's really nice about our platform is that we can independently control some of these variables of degradation rate, maybe like the size or the structure of the pores or the holes within the material that the cells can crawl in and around. And then also tune the composition of the material. So by adding different components like a growth factor or some sort of other cell signaling bioactive component to kind of help the cells in that environment following implantation to kind of not do the wrong thing. But I think that's a major challenge in this field is really understanding upon implantation, what are the cells in the local environment going to do? And so we think that by utilizing these silk materials and by including the appropriate bioactive factors, maybe we can limit the potential for chronic inflammation and scar tissue formation through the ability to slowly release things that might kind of trick the immune system into not responding in a negative way that would not promote wound healing in these tissues.
Do you have to use man-made materials, at least for the scaffolding, or
Yeah.
I guess what I'm asking is, how do the cells know? How do the cells signaling works so that the tissue knows where to go, how far to go, the pore size, and all that? Like, how does the body and the cells do all this without any instruction from outside?
Yeah, so that's a very great question as well. Often termed the fetal gene reprogramming or fetal gene expression. And so when you're right, during development as a human, we express certain genes and our cells secrete certain proteins, and they kind of follow this unknown and really awesome track in order to generate a full human. And so recently, people have shown through the development of organoids and things like that, utilizing stem cells, that cells that haven't fully matured or that have been kind of backwards programmed to have more of a fetal gene-like expression are actually able to regenerate structures based on local cues from the environment. And so the trick that every biomaterials and tissue engineer hopes to be able to find is what exactly is that appropriate set of environmental and local microenvironment, the cues, what is that appropriate set of cues, both ones that are stable and stuck, like the material, versus soluble ones that are floating around kind of in the liquid. How do we find this perfect set of cues, and how do we know over what time frame those sets of cues should be expressed? And so we're really hoping to answer some of these questions because they're not known. The method or how to do this is really not fully known, and it's going to be different for every tissue type. And so by utilizing cells that are not fully mature, we have some ability to just get them to do what they know how to do and become mature cells. However, in patients who have diseases like muscular dystrophies or things like this, where the cells are already doing the wrong thing, then we can't expect the cells to just kind of go about their business and form nice new tissue. And so it's really important that we do both the clinical and implantation or in vivo studies to look at responses to these materials alongside fundamental studies in the lab, like in a dish, where we can begin to understand how the cue that we give our cells changes their long-term behavior almost. And so we're really focused on understanding the main parameter of time within tissue regeneration and how we can utilize our materials to adjust time-dependent cues during the wound healing process. And so from a biomaterials standpoint, these are things we control with how we build the scaffold, how big the scaffold is, how cross-linked it is, the structure of the scaffold, how elastic it is, these types of things. But in all situations, every time you change a cell type, even if you use, some muscle cells from a pig or a rat, the response is going to be different than if you use muscle cells from a human. So every creature is slightly different. And so, it's important for us to try to answer these questions prior to just sticking this material in a human and hoping that it works. And so I think that's right. Kind of what makes this field take a long time to reach a clinical realization of a material in vivo.
Yeah, and then what percentage of a given tissue is scaffolding and man-made materials versus the cells that innervate it and grow around it?
Yeah, so for our scaffolds, they're kind of like a sponge. So we make these sponges out of silk from the cocoons of a silkworm. And so in most cases, the void fraction or the airspace prior to putting any tissue in there can be controlled based on how much polymer you add. And so in most cases, the scaffolds we're using right now for a lot of our skeletal muscle applications are about 65 to 75% scaffold, with the opposite, the 25 to 35% being open space where tissue can grow and form. However, because the protein or because the scaffold is made out of silk, which is a protein, once that scaffold is put into the body, enzymes that are secreted by cells can actually degrade the scaffold. So over time, the scaffold will disappear and be replaced completely by tissue. However, it's this rate of degradation that if it happens too fast, then the tissue may be too weak, or as you mentioned before, may not have enough vasculature or enough blood vessels in order to support the healthy maintenance of that tissue. And so we're really interested in this dynamic interplay between tissue infiltration into the material and then the deposition of new protein and healthy tissue formation without inflammation or with limited inflammation, and then kind of the slow degradation of the material as the new tissue that's been deposited integrates well with the injured area.
If the scaffolding disappears, though, why would there be an immune response? Or wouldn't there just be a temporary one that goes away?
So in a lot of cases, the patient that we're interested in treating has had a traumatic injury. And so, for a skeletal muscle repair, perhaps this could be a casualty of war or a car accident or something like that. And so anytime a patient has injuries or even surgery, all of these induce an immune response. And so an acute immune response or a short-term immune response is healthy and is good for wound healing. However, a chronic immune response will often lead to poor tissue formation and actually will end up replacing what should be healthy tissue with scar. And then in a lot of applications and clinical manifestations of these injuries, the scar tissue continues to expand over time. So instead of your injury healing into a healthy muscle following the implantation of some sort of scaffold or something like that, the tissue will form scar-like tissue, and instead that scar-like tissue over time will kind of expand and take over local healthy tissue that wasn't even injured in the first place. And this scar expansion, both in skeletal muscle and in cardiac muscle, actually is often what leads to basically decreases in patient prognosis or the ability of the patient to really use the muscle for its function. And so like a heart attack, for example, when a patient has a heart attack, there's scar tissue that forms at the site of the blocked blood vessel. And then because of the way our bodies are made, our cardiac tissue doesn't regrow, and our cardiac cells are unable to kind of repair this injury. And so what happens is the scar tissue where that original injury was will expand over time. And so for some patients, the scar will expand over a period of 10 years, and it won't necessarily influence their quality of life. But for other patients, it will expand much faster. And this is known as the transition from having, just having had a heart attack to actually transitioning into heart failure. And so the same thing is true in skeletal muscle. It's just not as catastrophic for the patient.
Well, when you say the scar expands, does it co-opt normal tissues and turn them to scar, or it just grows and pushes out normal tissue?
Yeah, so it actually takes over normal tissue, and the structure of the normal tissue turns into the structure of scar tissue. So the cells that are there kind of take on an angry or inflammatory mindset and change their gene expression and actually make proteins and things that are representative of scar tissue. And so what at the time of injury was actually healthy tissue over time turns into scar tissue. And this is actually a major clinical problem and something that we're hoping our materials are able to address in the future.
Yeah, that's very odd. I mean, how small of a nucleus or a site or a seed of scar tissue is needed to have it proliferate and eat up all the surrounding tissue?
Yeah, so I wish I knew the answer to your question. That would make my math and material design a lot easier. Unfortunately, I think that that's really going to be patient to patient dependent. In the same way that a cold influences one patient's immune system differently than another's, these types of inflammatory events at the muscular level influence different patients in different ways. So characteristically, children are much better at recovering from injury than elderly adults. And as I mentioned before, patients with diabetes are also less adept at recovering from injury than patients who are the same age but are not diabetic. And so we're hoping that we can start by trying to classify our patients based on risk factors and from their environment and from their known medical history, so that we can develop a platform of materials that will kind of suit a patient under a certain set of risk factors. So hopefully a surgeon would be able to go in and say, okay, I'm going to use a cell-free material to augment this injury and help this patient recover. However, we anticipate that the wound healing rate for this patient will be slower, maybe over a couple months. And so we're going to choose this product. And, if there was also a child in comparison, maybe they would choose a different product that had a faster rate of degradation and release of factors that promote wound healing in comparison to what product they might choose for an elderly patient with a similar injury.
Okay, yeah, that all makes sense. It's just why we don't know why so many questions. But another thought that came to mind is, what if you created a tissue with a scaffolding where the scaffolding was internal to the tissue? There was at least one or more layers of cells that comprised the outside of the tissue, and the scaffolding was purely internal. Would that be enough to hide it from the immune system, yet still provide enough structure for the tissue to survive until the point where the inner scaffolding gets eaten away or taken away?
Yeah, so this strategy is actually, or the strategy of at least hiding the material or hiding the cells from the immune system, is really common in kind of the tissue engineering field for type 1 diabetes, where there is kind of an immune response. The reason a patient has type 1 diabetes is their immune system attacks their pancreas. And so there's a lot of effort to make materials that hide from the immune system for applications for type 1 diabetes. However, I think for us, in the idea of treating skeletal muscle, we actually want our materials to stimulate the immune system at the right way and at the right time. So this is basically the fundamental nature of our work. We are trying to understand what that right time is and exactly what the right way is. And so instead, if we can harness the power and the wound healing power of our immune systems, then we should be able to make these materials that actually improve wound healing rather than just supplement or fill gaps or things like that, which is what our current materials do. So instead, we're interested in building materials that can control the wound healing environment and kind of bring the cells that are causing scar tissue formation, the proteins that they're secreting that make scar, instead trick them almost into making proteins that are the basis and structure for healthy tissue.
What about the scarring process itself? Has this been studied extensively? And what's causing the scarring? Is it the immune system, trying to surround and block out the offending elements, or is it the cells themselves that are changing, that are just next to the to the foreign element, and those are forming around it? Like, how does scarring happen?
Yeah, so in for example, in skeletal muscle, there are numerous different types of cells that are present within the tissue. There's the actual muscle cells themselves that allow you to run or to pick up your water bottle. And then there are the vessel cells, so endothelial cells and smooth muscle cells that kind of make up your blood vessels and the structures of those. In your muscles, you also have nerves, which is how your brain communicates with your leg and tells it to move. But then you have this other kind of devious population of cells that are termed in some cases fibroblastic, in other cases stromal cells, and they're basically like a filler cell population. And in a healthy patient where there is no wound and no inflammation, these cells are kind of quiescent, or they're not doing very much. They're just kind of there for maintenance and help you just maintain your muscle the way that it should be. However, when an injury happens, the fibroblasts and stromal cells that are present within the tissue are really important for example, in making clots. So fibrin is a main component of clots, and fibroblasts or the stromal cell population can be a component of clot formation and secreting the right proteins to kind of prevent you from bleeding in lots of places. And so this stromal population, which includes these fibroblasts, are really the main component of scar tissue formation. So fibroblasts or what is termed a stromal population are known for making extracellular matrix. So the proteins that kind of make up the bulk of your tissue. And so this population, when injured, will make new extracellular matrix. And when wound healing is perfect, it will, you'll generate new extracellular matrix, muscle will regrow, and everything will be just fine. But in situations where wound healing isn't perfect, the fibroblasts will get, I guess, almost overstressed, and they'll start making collagens and start making these thick fibers that are kind of unaligned and not properly oriented within the muscle tissue. And that's what starts to generate scar. And so really, scar tissue is great because it keeps your tissues from falling apart. But the expansion of that scar tissue is what can lead to kind of negative patient prognosis. And so we want to use our materials to kind of control and trick this stromal population and then the local immune cell population that's present within the tissue into not expanding the scar when it's not necessary.
Inside the body, when there's a scar, is it homogeneous or is it heterogeneous? Does it have like different layers with different types of cells in it, or is it just all one thing that just keeps growing outwards with no different surface on it or interior?
Yeah, so scar tissue, I would say, in comparison to healthy tissue in almost any organ, is very random. And so I'm not quite sure how the body does it, but the body is very good about organizing its cells, creating tissues that have different structures. Your muscle is very anisotropic, or it has very long muscle fibers that form within the muscle. And when scar tissue forms, it goes in every which direction. It doesn't have that local kind of organization that your healthy tissue would. I mean, even in the liver or in your skin, the scar tissue doesn't form in the same structure as the tissue that should be there. And so usually this mismatch in kind of the mechanical properties of the tissue, so how elastic the tissue is, for example, and a mismatch in kind of the composition, so the chemical composition of that environment continues to engage the local stromal population and anger it in a way that then leads it to continue to make scar tissue. However, if we knew how to stop the stromal population and the fibroblasts from making more scar tissue, we would be doing it, but we don't. unfortunately.
Right.
And in
What
every
about
tissue,
when scarring ends on its own, when it stops on its own accord? Does it tend to form an outer layer that's different from all the other layers? Maybe that's the signal that tells the body, okay, we scarred enough, stop.
yeah, so I think what really happens in situations like that, where scar tissue does not end up hurting or impeding final tissue function, is that inflammation kind of settles down and you don't continue to have the local bioactive signals in that area that promote more scar tissue formation. And so in different tissues, in say skeletal muscle versus the liver, for example, those bioactive cues are going to be different. And as biologists and tissue engineers, we aren't quite certain about which factors are showing up at exactly which time and how to mitigate their existence.
When with any implant in the body, then, is it safe to say that scarification will always happen and it'll continue ad infinitum, or does it stop with any man-made material from inside the body? You know, a pacemaker, a hip implant, whatever kind of implant it is.
Yeah, so a lot of times there are things we can do to alter scar tissue formation. So things like anti-inflammatories, when given at the right time and for, and at the right dosage, can help and prevent scar tissue formation. But hip implants, for example, we consider a lot of times the composition of those implants to kind of be ones that are designed to hide from the immune system, and thus they're not as likely to induce scar tissue formation as compared to, say, one of these more natural materials where we want new tissue to grow in its place. And so for a hip implant, we're not asking new tissue to grow, and we're not asking new bone to form. We're just putting a foreign object there in replacing that tissue. And so I think the main difference in between, say, a pacemaker or a hip implant and some of these newer tissue engineered strategies are that we're really trying to regrow or rehabilitate the tissue at the injury site as opposed to fully replace it with a foreign object.
So what does the body use to hold together your liver or to hold together your other organs? What kind of matrix scaffolding does it use?
So the body makes its own scaffolding, and that's termed extracellular matrix, which is a combination of different proteins like fibronectins, collagens, laminins, periostins, which are all different kind of, you could think of them as like biopolymer type proteins that are long and can intertwine with each other, can cross-link like any sort of synthetic polymer, but they have different sites along the structure where cells can grab on. And so this happens through integrin binding. So cells have integrins on the surface of their cell membrane, and these proteins will recognize different protein sequences in the extracellular matrix and essentially grab on like a handle. And so this is how cells kind of sense their environment. And so by utilizing natural materials or extracellular matrix-based materials within our scaffolds, we can promote cell attachment and cell adhesion and things like that within our systems. However, natural materials that come from non-mammalian sources, such as silk protein or alginate, which comes from sea kelp or brown algae, these natural biopolymers don't actually have the handles on them that allow cells to grab on. And so a lot of times when we're utilizing these natural materials that come from non-mammalian sources, we have to engineer handles onto these biopolymers so that our cells can grab on, actually.
Why can't we use the extracellular matrix as the scaffolding? Is it just hideously complex that we isolated the different components and tried to grow a matrix from them?
So actually we do. We do use extracellular matrix as scaffolds. And so there's a really awesome company called Ventric that is out of California, which came out of Karen Chrisman's lab at UCSD. And this company, Ventric, has a clinical trial that's ongoing that utilizes natural extracellular matrix from pig hearts. So the pig hearts are decellularized and all the cells are removed. And then the extracellular matrix components are kept and utilized in order to make a scaffold for applications in the heart. And so this is really awesome. However, in this type of situation, we have no control over the degradation rate of the material. We're kind of stuck with what the body wants to do. And so through combinations of materials like silk or alginate and extracellular matrix components, we can add extra engineering parameters such as ability to tune degradation rate in vivo that we wouldn't have if we only use kind of naturally derived extracellular matrix components.
Okay. Well, I learned a lot. You certainly have a difficult task ahead of you, but I appreciate all your insight. So what's the best way for folks to learn more and to maybe see some of the work? Where can they go?
So you can definitely check out my website, which is www.stoppellab.com. But then I also like to send students and my family to check out, there's a journal called the Journal of Visualized Experiments, JOVE, and you can find their stuff at jove.com, I believe. And the JOVE website actually takes research that has been done by people in their labs at universities and makes short videos that describe the research they've done. And so there's some great JOVE articles on natural biomaterials and decellularization that I would highly recommend you check out.
Oh, very cool. Very, very excellent. Okay. Yeah, I can see on the site they have nearly 10,000 video articles. Wow.
Yeah. So it makes it really nice to kind of translate vocabulary that is often foreign to those who are new to the topic into something they can visually see.
Excellent. Well, that's great. Well, Whitney, I appreciate you coming on the podcast, and thanks for your time and attention and everything.
Yeah, thank you so much.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.